Hello and welcome to this discussion on SHAPE, or selective 2'-hydroxyl acylation analyzed by primer extension, or SHAPE for short. So SHAPE is utilized for RNA structure determination. We use SHAPE to determine the RNA structure of a given molecule of interest. Now, in terms of RNA structures, RNA can form secondary RNA structure and it can also form tertiary interactions. In terms of the secondary RNA structure, we can deduce the secondary RNA structure using the RNA sequence only. We can do that via energetics. We can also do that using sequence alignment, or we can do that with any of the previous methods that I just mentioned, plus using extra experimental data that we can get via DMS probing or SHAPE probing. And SHAPE is what I'll be talking about in this module. Now, in terms of tertiary interactions, we can deduce them via crystallography and NMR studies, but this cannot be applied for all RNA structures, so this has to be kept in mind. So the question becomes, what is the overall principle of SHAPE? SHAPE relies on the reactivity of the 2'-hydroxyl, so let me discuss that. So the principle of SHAPE is that it relies on the reactivity of the 2'-hydroxyl. So within an RNA structure, conformationally flexible nucleotides can sample multiple conformations. And so a change in the flexible nucleotide conformation can alter the 2'-hydroxyl reactivity. Here to the right, you can see two ribonucleotides bonded together via a phosphodiester bond. And what you can see is the 2'-hydroxyl. SHAPE relies on the reactivity of this 2'-hydroxyl. Now, the 2'-hydroxyl makes the RNA molecule more reactive, and this is something that we're going to utilize in SHAPE. But what you have to keep in mind is certain nucleotides are flexible and they can undergo certain conformations that make it so that that 2'-hydroxyl is more reactive, relatively speaking, to other nucleotides or to other RNA nucleotides within that same RNA molecule. So keep in mind that the 2'-hydroxyl's reactivity is influenced by the local nucleotide environment, and some conformations will result in an increase in the 2'-hydroxyl nucleophilicity. Now, factors that influence the 2'-hydroxyl's reactivity are a few. For example, electrostatic interactions of the 2'-hydroxyl along with the 3' phosphodiester can make it so that there is an increase in the nucleophilicity of the 2'-hydroxyl. So, for example, if you have the 3' phosphodiester non-bridging oxygen atoms, these right here are the non-bridging oxygen atoms. If you have those pointing away from the 2'-hydroxyl, then this would increase the 2'-hydroxyl's reactivity. There are other influences, and you can check the papers cited below for other ways in which the 2'-hydroxyl's reactivity can be increased depending on certain interactions between the 2'-hydroxyl and the environment. Now, in terms of flexible nucleotides, I've mentioned this term, and what you should recognize is that flexible nucleotides tend to be single-stranded, and flexible nucleotides have more potential to undergo conformations that increase the 2'-hydroxyl's reactivity. It's the local environment that influences that 2'-hydroxyl, and it is the factor that can make it so that that 2'-hydroxyl is more nucleophilic. And then when we react it with certain electrophiles, then we can deduce the reactivities of the different 2'-hydroxyls of an RNA molecule. And then we can use that information along with bioinformatics, for example, or a bioinformatics software to deduce the overall secondary RNA structure with increased information that we've obtained from the SHAPE. Just as we had flexible nucleotides, we have constrained nucleotides. These tend to be base paired, and they tend to be engaged in constraining tertiary interactions. Now, you have to keep in mind that there is no relation here to solvent accessibility. Now, nucleotides can be unconstrained even if they're deeply buried within a complex RNA tertiary structure. And constrained nucleotides have less potential to undergo conformations with increased 2'-hydroxyl's reactivity. So it is the flexible nucleotides that have the potential to undergo certain conformations, and those certain conformations can have increased 2'-hydroxyl reactivities or nucleophilicity, and then we can use those reactivities, we can use that information to deduce the secondary RNA structure. So the basic premise of SHAPE has been outlined. Basically, the 2' hydroxyl's reactivity is influenced by the local nucleotide environment, and thus probing the 2' hydroxyl's reactivity within an RNA structure provides insights to that RNA structure. The SHAPE reagents that I mentioned earlier are electrophiles. These electrophiles are typically anhydrides and acylcyanides. For example, isotoic anhydride is a SHAPE reagent often used, including N-methyl isotoic anhydride is another one, and another one would be 1-methyl-7-nitroisotoic anhydride, or 1M7 for short, along with benzoyl cyanide. Now, what you should keep in mind is that those SHAPE reagents have different uses. So some SHAPE reagents can be used in cell-free studies and others can be used otherwise. And so you have to keep in mind, as you can see on the figure to the right, is that different SHAPE reagents have slightly different uses in some cases. And so depending on your optimization parameters, you can alter the protocol of your experiment so that it is suitable for your particular project. So what SHAPE reagents do is they form 2' oxygen adducts. And so the 2' hydroxyl of conformationally flexible RNA nucleotides will react with electrophilic SHAPE reagents to form 2' oxygen adducts. This is basically 2' hydroxyl acylation. So you can see on the figure at the bottom that you have a constrained conformation of a particular RNA with the 2' hydroxyl in red, and you can then see a flexible conformation. They go back and forth, not necessarily in complete equilibrium, but they can go back and forth. And what you should keep in mind is that when you introduce the SHAPE reagent, there's a potential that that SHAPE reagent will react with particular 2' hydroxyls within the RNA molecule, and that will form 2' oxygen adducts, and we'll use the location of those 2' oxygen adducts, or the locations of those 2' oxygen adducts, to deduce the overall secondary RNA structure. And we'll also utilize the reactivity information to deduce the overall secondary structure. So in SHAPE, reaction conditions are optimized. I've mentioned optimization earlier very briefly, but the reaction conditions are optimized so that there is 1 to 100 to 1 to 300 probability of forming 2' oxygen adducts at any given nucleotide. And so the modifications are scattered and few. You won't be seeing those modifications everywhere within every single 2' hydroxyl group within your RNA molecule. Rather, what you'll have is just sort of scattered and few 2' oxygen adducts that are being formed. And we'll use those to determine the overall secondary RNA structure. So reverse transcriptase mediated primer extension, as you can see in the name of this slide, is the way in which we detect the locations of the 2' oxygen adducts. So what we do is we perform reverse transcription of the RNA with the 2' oxygen adducts. This is the RNA that has been modified and has been introduced to SHAPE reagents. But we also have to have a control, and that control is basically where we do reverse transcription of the same RNA molecule, but it is an unmodified RNA molecule. That is, it has not been introduced to SHAPE reagents. And this will help locate the control, that is, sites of natural reverse transcriptase pausing points. And so sometimes the reverse transcriptase is going to pause, and we don't want to think that that pausing point is because of a 2' oxygen adduct when there's actually no 2' oxygen adduct there. And so another reason is because it can locate sites of RNA degradation. Now, reverse transcription is halted at one nucleotide prior to a 2' oxygen adduct. And so the cDNA lengths will help locate those adducts. When you do reverse transcription, you get the cDNA, obviously, and then that will help locate those 2' oxygen adducts that have been formed. Now, the typical way that SHAPE is done is through the use of capillary electrophoresis. Here to the figure on the right, you can see a couple of important things. On the very top, you can see 2' oxygen adducts in red. These have been formed on a given RNA molecules that you can see in the figure, and they form at different sites, as you can see here. So those sites is where you had a 2' hydroxyl that was relatively flexible, that had a conformation whereby the 2' hydroxyl was more reactive, and it reacted with the shape reagent or with the shape electrophile that you introduce, regardless of what electrophile you use. Now, obviously, you have to use the right electrophile with the right shape reagents for your particular needs, whether you're doing cell-free studies or otherwise. You have to keep that in mind. We then do reverse transcription. So in green, you can see the reverse transcribed cDNA. And then that reverse transcribed cDNA will stop one nucleotide prior to a 2' oxygen adduct. And so by this, we can locate the exact location of those 2' oxygen adducts. A capillary electrophoresis electropherogram is typically the graph that you're going to get, and in a capillary electrophoresis shape. Now, some people do shape in other means using other ways. Now, here you can see the positive reaction or the positive or the actual experiment whereby shape reagents were introduced into the RNA molecule of interest. And you can see that in blue. In black, you can see the negative control, and that's going to help locate natural pausing points of reverse transcriptase and sites of RNA degradation. And the way we deduce the shape reactivities is basically by more or less subtracting the fluorescence of the reactions whereby we introduced the shape reagents and the reactions whereby we did not introduce any shape reagents, the negative control, that is. And you can see on the graph in the figure that there is also sequencing that has been performed. So sequencing is utilized so that you can know exactly where those shape reactivities are occurring, or the nucleotide position of those shape reactivities. Now, capillary electrophoresis is an analytical method. Oftentimes, there are preparative techniques for nucleic acid separation that we're familiar with, like polyacrylamide and agarose gel electrophoresis. And capillary electrophoresis is very similar in concept, except you're utilizing very small quantities of DNA that can be loaded onto a capillary, and then you're performing PCR after separation. And so what you get is a CE electropherogram, as you can see in the figure at the bottom right. And that CE electropherogram is what's going to be used, generally via software, to deduce the reactivity profiles of shape. Now, in terms of cDNA sequence, shape also involves sequencing of the cDNA products. So in practice, instruments that contain an automated DNA sequencer and a capillary electrophoresis will do this kind of simultaneously. So the same machine will do two things. Now, fluorescent intensities of the shape reagent reactions and the control, whereby you had no shape reagent reactions, are aligned with the obtained RNA nucleotide sequence. There are various available softwares that can be used to perform shape, and those include ShapeFinder, ShapeCE, and QShape. Now, nucleotides reactivity profiles are generated by subtracting the intensity of modified RNA peaks from the intensity of the control peaks, or the background noise. Remember, we mentioned the natural pausing points of reverse transcriptase and sites of RNA degradation. And there are various normalization methods utilized by these softwares that are utilized in these softwares, like the alpha parameter and other probabilistic models. Some of the cited papers below discuss these things, although this is generally a software feature that you don't have to worry about. So I want to delve deeper into shape and offer some further information that can elucidate what shape is in a broader perspective. So the first thing that we mentioned is that shape is relied on to deduce the secondary structure of an RNA molecule. It's relied on to deduce the secondary structure of an RNA molecule when combined with energetics, as I said earlier at the beginning of this module. The first thing to recognize here is that determining the base pairing pattern, or the secondary structure, is an easier endeavor than that of determining the 3D structure. Not all RNAs are appropriate candidates for things like crystallography and NMR studies. So this makes it so that you can't always predict the 3D structure of an RNA molecule. Now, determining the secondary structure, irrespective of the 3D structure or the tertiary interaction, is possible because hydrogen bonding and stacking interactions are stronger than tertiary interactions. So you have to keep in mind that you start off with an RNA sequence, a linear sequence, and then that linear sequence will undergo some kind of base pairing. So we can just say that you have some kind of base pairing. I'm just drawing random base pairing here. And so you have some kind of base pairing. And then you have further on some tertiary interactions that can happen. Now, determining the tertiary interactions is difficult because not all RNAs are candidates for things like crystallography and NMR studies. Determining the secondary structure is a much simpler task. And the way we do this is we combine information from energetics. So we use the sequence and we use nearest neighbor thermodynamics along with algorithms like dynamic programming to deduce the secondary RNA structure, and we combine this with information that we obtained from shape. So remember, with shape, you have certain things that are, or certain local environmental conditions that make a particular nucleotide a flexible nucleotide and can react with a shape electrophile, and that makes it possible to obtain some information regarding the secondary structure. So for example, in loops, you tend to have flexible nucleotides, and also when you have single-stranded regions, you tend to have flexible nucleotides, and we can use the shape reactivities to kind of supplement the energetics so that we can deduce the overall secondary RNA structure with higher accuracy up to over 90 or up to 95% or greater accuracy in deducing the secondary RNA structure. So the reason we can do this, the reason we can deduce the secondary RNA structure irrespective of the 3D structure, has to do with the hierarchical nature of RNA folding. So RNA folding is sequential, it's hierarchical. Secondary structures form prior to tertiary structures. So you form the secondary structure prior to the tertiary structures, and it happens to be that those hydrogen bondings that you see, along with the stacking interactions, being stronger than the tertiary interactions that happen in the 3D structure. And so this makes it possible to predict the secondary RNA structure irrespective of the tertiary interactions. Now, we can attain a glimpse of a potential secondary structure of a potential RNA molecule using sequence information alone or in combination with sequence alignment. And this can provide information regarding the secondary structure of RNA, but you want to constrain that because that's not very accurate. The way you constrain that is by adding information that you obtained from shape. And you can do this via software, and so that can be done automatically for you in a way. Or alternatively, some people do this manually. Now, sequence-based folding relies on energetics and thermodynamic parameters. It also relies on algorithmic models. We'll talk about that in a bit, so that you can deduce the secondary structure. But recall, this alone would not be accurate, so you'd have decreased accuracy in the secondary RNA structure that you've deduced. Now, in terms of energetics, the basic principle here relies on nearest neighbor parameters. These information are obtained from optical melting experiments. And the principle here is that the structure that forms the lowest free energy is likely to be the one that's biologically active. Now, dynamic programming refers to algorithmic models. These are deterministic models, deterministic algorithms with predefined energetic rules that can be utilized to obtain or to deduce a potential secondary RNA structure. It finds the lowest free energy structure, but you have to keep in mind that this alone, without using shape information, would not be very accurate. So the idea is to combine energetics and shape, so the secondary structure can be elucidated with high accuracy, equal to or greater than 95% accuracy. And by combining the thermodynamics-based dynamic programming that I've mentioned earlier and shape. And so you can combine those so you can get a very accurate potential RNA secondary structure.